And there'll be 5 talks that follow and that focus on a wide spectrum of seafloor habitats with an emphasis on benthic flux feedbacks to the water column. So in this introductory presentation, I'm just going to try to paint some big picture perspectives on the role of seafloor sediments in ocean carbon and biogeochemical cycles. I'm going to talk about how global fluxes of organic carbon to and from the seafloor have been assessed, kind of giving a quick recap of how we've made some advances in this area over the last 40 years. And then I'm going to introduce a new technique for determining benthic fluxes that is becoming popular. It's called eddy covariance. It's a great method in that it allows you to look at the dynamics of fluxes, particularly under natural flow and light conditions. And 2 of our speakers will highlight work using this method. Okay, for our big picture, we're going to start by just reminding you what Earth's hypsographic curve looks like. And the reminder, of course, that some 70% of the Earth's surface is covered by the ocean. But what that means is that some 70% of the Earth's geological surface is the seafloor. And that seafloor ranges in depth from, of course, the shoreline out to the ocean trenches, which we'll hear about today, that exceed 11,000 meters. The mean depth of the ocean is 3,729 meters, and that's roughly 2 times the deepest part of the Grand Canyon. So I think it's useful to, again, start thinking about the seafloor, to think about what the Earth would look like if we looked down from space and could see through all that water to the seafloor and see the wide expanse of seafloor sediments that cover, again, 70% of the Earth's surface. So we would see a range of compositions. We would see that snow line that Bernie talked about on ocean ridges, and we'd see changes as we come up on the margins. But what does the seafloor really look like? We've only learned that in recent years by using camera systems and ROVs. And most of what we learned before that about the seafloor was from sediment cores. But here's a little video showing you the seafloor. This is from a Bissell station, Station M, which Ken Smith at Mbari has studied for over 30 years, looking at the seasonality of fluxes in a deep sea environment. You can see hemipelagic sediments, rich marine life, a lot of holothurians and fecal casts and that sort of thing. Again, a lot of spatial variability even in this deep part of the ocean. The video here is going to transition now to another area I've been interested in called Hydrate Ridge off the Oregon coast. Notice the rain of particulate matter that comes to the seafloor, particularly near a productive margin. And we find chemosynthetic communities on the seafloor. These are some bacterial mats forming over an area where there are gas hydrates just below the sea surface. Or we can move on to the shelf, where we've been studying the Oregon shelf. We have sandy sediments. This is at 80 meters depth, still highly bioturbated. In winter months, when you have storms, you see sand ripples. Again, there's a wide variety of habitats and sediment types on the seafloor. And again, most of what we know about the seafloor comes from taking sediment cores. If you ever wondered what happens when you send one of those things to the seafloor, here's just a video using a system that we use for our studies on the Oregon shelf. It takes essentially a gravity core that pulls up a nice about 50 centimeter sand core that we bring back for characterization and core incubations. Again, so that was just to try to get you enthusiastic about the seafloor. Now, again, I mentioned most of what we know about seafloor biogeochemistry has come from collecting sediment cores and having archives of these cores. And this is a figure from a nice paper that was published in Geology in 2015, showing the lithologies of surface sediments globally based on over 14,000 core samples. And the authors of this paper, who are from the University of Sydney in Australia, make a nice point that the surface distributions of lithologies and compositions and accumulation rates of sediments really helps us understand, gives us an underpinning for understanding ocean biogeochemical cycles and sediment transport in the ocean. Of course, cores are also our window into the past through downcore studies of the paleoceanographic record and past patterns of ocean primary production. In this figure, for instance, you can see, if you look at the sort of blue colors, the calcareous oozes that, again, are on the ocean ridges along the equatorial Pacific, large parts of the Indian Ocean, and then the siliceous oozes that ring, for instance, the Southern Ocean. So in this session, again, we're going to cover a number of habitats, but there's some key points that we'd like to stress about the roles of seafloor sediments in biogeochemical cycles. The first is that this interface is an interface between the geosphere and the hydrosphere, and it's a reservoir that's central to the mitigation of ocean acidification. We heard about that in this morning's session, and that's particularly through dissolution of sedimentary calcium carbonate. But as Tom Bianchi reminded us yesterday, sediments, particularly on margins and in areas like deltas, or fjords, are areas where there's carbon sequestration through burial. Sediments, seafloor is also a mixing zone for terrestrial and marine organic carbon, and this can lead to high rates of both aerobic and anaerobic respiration. And this can then lead to the return of DIC to the water column and its export to the open ocean from margins. In our first session of this meeting, we heard about ocean deoxygenation and hypoxia. Hypoxic areas are tied to these areas of high respiration. And these are also areas, of course, where we have inorganic nitrogen losses by denitrification, trace metal redox speciation changes in fluxes, scavenging, and orthogenic mineral formation. Another reason to think about the seafloor is this is a zone of gas hydrate formation and dissociation. And Katie will be talking about this process in the Arctic. And then, of course, last but not least, the seafloor is a vast habitat for life and for metabolism, and it spans, of course, from the shallowest depths to the deepest depths of the ocean continuum. So in preparing for this session, I put together this figure is to try to compile the role and sort of the relative magnitudes of fluxes to the seafloor in the carbon cycle compared to other parts of the carbon cycle. And we could debate a lot the numbers, but I want you to sort of look at some of the relative magnitudes. And this is based on what's considered the present day oceanic carbon cycle. So by present day, you can see in this figure that the CO2 flux, the net flux into the ocean from the atmosphere is, it is into the ocean in both the coastal and the open ocean areas. It's portrayed that way. But I've also tried to highlight fluxes to the seafloor and tried to separate, again, the sort of upper part of the ocean above 1,000 meters, or 1,000 meters, and the deep ocean. And one of the reasons for that is that when carbon reaches the deep ocean, either if it's buried or even remineralized in this zone, at least it's sequestered from the atmosphere on human timescales. So we are interested in what fluxes do come down through the biological pump to this realm. And so what I wanted to walk you through now is just how we have some estimates for the magnitudes of those fluxes. Will Berelson said, I think at one point, how do you put a number on some of these things? So I want to show you how we put one number on this figure. It's the carbon flux to the seafloor below 1,000 meters, and I'm talking now about the organic carbon flux. And I'm just a reminder, I'm talking about 88% of the ocean when I talk about the seafloor below 1,000 meters. So how have we come to know the magnitude of this flux, which again is a carbon flux that is again removing carbon from immediate contact with the atmosphere? Well, our first estimates of this flux really stem from programs that started in the 1970s where sediment traps were put out globally to measure the sinking flux. Shown here in this diagram is milligrams per meter squared per day as a function of depth. The very first compilation of such sediment traps fluxes was made by Erwin Suess, who happened to be my thesis advisor. That's why this one was drilled into me, I guess you might say. But the observation that there was an exponential decrease in carbon flux with depth in the ocean, but that it scaled with the net primary production at the surface. The Suess relationship was based on the data that's shown with the solid symbols in this diagram, which is from a later paper by Lutz et al. Of course, there's been a lot more data that's been added, and a lot of this in this figure came from the Jagoff's program. But you know, and so we've had revisions of these exponentially decreasing relationships, but the general model stays the same, and the thought that carbon drops off as a function of depth and degradation rate and settling rate of particulates. So just to remind you, then this kind of relationship was used by Mueller and Karger et al. In 2005, combined with assessments of net primary production that's based on satellite information and models of chlorophyll, PAR, and sea surface temperature, and also then ocean bathymetry, to come up with, again, based on the sinking flux, a global distribution for the POC flux to the seafloor. And if you integrate this figure, which they produced, for the areas of the ocean below 1,000 meters, the global flux is 0.31 petagrams of carbon per year. And for the margins, they get about twice that, or 0.6 petagrams carbon per year. Well, there's another approach to carbon fluxes, which gives us a slightly different answer. And that's based on looking at the seafloor, because the seafloor is our ultimate sediment trap. And one advantage of the seafloor is that, unlike sediment traps, you're going to see lateral inputs, particularly near the margins. And sediment traps may miss those inputs. So again, the idea to assess a flux to the seafloor, and it could be for carbon or silic or any part of the biogeochemical cycles, is that the flux to the seabed has to be equal to the sum of what is remineralized and what gets buried. And in the case of organic carbon in the deep ocean, almost all of the flux is remineralized. So if we measure that remineralization rate, we have a good measure of the carbon flux to the seafloor. And so one approach to that is to measure the oxygen flux, because in the deep ocean, most of carbon is oxidized through aerobic respiration. So we use oxygen fluxes as a proxy for carbon fluxes. Briefly, until recently, there were two main methods for arriving at oxygen fluxes. And again, these developed in the last few decades. The first is based on modeling pore water gradients. And in case of oxygen, we were able to measure highly resolved pore water gradients using microelectrode sensors that can be deployed in situ to measure gradients across the sediment-water interface. Here's an example from some of my work off Oregon showing oxygen gradients profiles that only extend a few millimeters into the sediments on the Oregon shelf. But again, based on that change of oxygen and the assumption that the transport of oxygen into the bed is dominated by molecular diffusion, fluxes can be calculated as diffusive oxygen utilization rates, or DOU. The other approach, again, which can be done in situ, is to put a benthic chamber over the seafloor, enclose an area of the seafloor, incubate it, and observe the uptake of oxygen or the release of CO2 or the release of nutrients. And this is another technique. It does not assume that the only transport mechanism is molecular diffusion. So it's termed the total oxygen utilization. And generally we see in the coastal ocean, total oxygen utilization rates do exceed diffusive oxygen utilization rates because of the influence of bioturbation and bioirrigation. Okay, I'm running out of time quickly. So the way we got to a global estimate was to, this was recognized by Wei-Jun Tsai and myself, that there seemed to be a relationship between DOU and sediment, organic carbon, and bottom water oxygen. You know, the two reactants, carbon and oxygen, and so one could form empirical relationships between these parameters. We developed one first for the Northeast Pacific, and then following work by Sider et al., based on a lot of later data, largely measured by Ronnie Glood's group, they put together other empirical relationships, these multiple regressions for 11 regions, and again came up with sort of global estimates of the particulate organic carbon flux to the seafloor, which again could be integrated to give us a global flux as 0.5 petagrams of carbon per year. I just wanted to say that the data behind all this is still sparse. More is needed. And one question we can ask is are these fluxes changing? As we've heard, the biological pump is changing, calcification rates are changing. So how is that reflected in fluxes to the seafloor? All right, so just a real quick, I guess I'm going to run out of time. Eddy covariance is a new method for assessing benthic fluxes. It's being used primarily for measuring oxygen fluxes. It requires two sets of sensors, an acoustic Doppler velocimeter that measures velocities, and in the case of oxygen, and some kind of oxygen sensor. And this picture shows a fiber optic oxygen sensor together with a thermistor. It's what we would call a waterside approach to flux measurements, because you're measuring velocities and oxygen usually about 15 to 30 centimeters above the bed in a rapid time series, which then gets separated into average and variation components. So again, we make these measurements usually at 64 times a second, 64 hertz. This is an example of just 60 seconds of data, and this is what it might look like. But to again arrive at a flux, we have to take a few steps. We need to separate the variations from a mean. And here there's just a short-term running average put through the data to illustrate the separations from the mean. But what's key here is, I just want you to notice, is that there are parts of the record, for instance, where the vertical velocity, that's W, is mostly towards the bed. And in those periods of the record, you tend to see higher oxygen concentrations. Whereas other periods, when the velocity is away from the bed, you have higher oxygen concentrations. And this is a case where the bed is an oxygen sink. And what you're seeing is the effect of eddies passing by the sensors. And eddies that have been in contact with the seafloor have a little less, those water parcels have a little less oxygen as they move up away from the bed than those that are moving down towards the bed. And so we take a time average of these variations to arrive at a flux. And as you'll see in the next talk, this can be used also to measure benthic primary production in areas where the seabed is an oxygen source, not a sink. And this is an example from a seagrass bed, paper by Hume et al. And again, time series data, these are, we tend to break into 15-minute segments. You can then look at the cumulative sum of W prime C prime. And basically the slopes of these curves gives you a flux. In this case, it's out of the bed, oxygen being a source. And then you can average these into hourly averages or daily averages. And so this is a new method for arriving at fluxes that, again, allows us to trace the dynamics with changing light, with changing currents, and is giving us a lot more information about these fluxes. So I have run out of time. I think we should move on to the talks that follow.